To those of you who are visiting New Orleans, welcome. Compared to what we just heard, I'm going to talk about the easy stuff, which is math and science and medicine. Okay, so my lab is interested in combating drug-resistant bacteria with a relatively new approach, right? So the problem with drug-resistant bacteria is there's, as we all know, there's increasing morbidity and mortality. Bacterial drug resistance is growing exponentially, and deaths are rising very rapidly, and it's predicted that the number of deaths from bacterial infections, of all things, is going to be and already is approaching one of the leading causes of death in certain populations, and so it's a growing problem. Conventional antibiotics are just not solving this problem. They failed from day one. Well, they're helpful, but they evoke resistance, and now we have organisms that have learned to resist essentially all conventional antibiotics. So we're taking a new approach to peptide antibiotics, which have been around a while, and we're at a relatively early stage. We have a lead, we have intellectual property. We're working on preclinical optimization, but we're far from the point of bringing this to the clinic, but this is our this is our goal. So we use synthetic molecular evolution to find the best peptide antibiotics. That is, we make rational peptide libraries. We screen them under clinically relevant conditions. We find gain of function sequences, and we typically do this iteratively. So we go through multiple generations of screening to find the best lead sequence. Now, this is an area, finding membrane active peptides for drug delivery, for antiviral activity, for antibacterial activity, where AI has failed. It's not currently amenable to machine learning or AI, to my knowledge. So, and we have gotten to the point where we're talking about a lead peptide. I'll abbreviate it Q7 here. I'll give you publications. I'll give you a QR code for publications at the end where this peptide is described in detail. But comparing our approach to discovering peptide antibiotics, we essentially show that under clinically relevant conditions, that is with host cells present in serum proteins and the sort of things you find in the body, most antimicrobial peptides, even ones like pexaganin here, which actually has been through several human clinical trials, not successfully, compared to these well-known antimicrobial peptides, or these well-known antimicrobial peptides essentially lose activity in vivo. They lose activity because they bind to host cells, they bind to serum proteins, and they bind to host tissue. So this is exactly what we screened against. That is, we screened for peptides that retain antimicrobial activity in the presence of concentrated host cells and tissue and serum proteins, et cetera. So we essentially screened under clinically relevant conditions. So we find peptides that we call host compatible that have broad spectrum activity against human pathogens. Okay, so against the so-called escape pathogens, these are really the leading multi-drug resistant, pan-drug resistant human pathogens we find in human patients. Our peptides, this lead peptide, for instance, has very good antimicrobial activity against all of them. So it's broad spectrum. We've shown in murine wound models that this peptide is effective at curing drug-resistant bacterial infections in wounds in animals, and this is kind of the market that we're thinking about. This is the application we're thinking about is wound infections, and including diabetic foot ulcers, which is a serious and growing problem. Infections in diabetic foot ulcers that often lead to amputation. So our peptides are very effective in wounds at treating drug-resistant bacteria, both gram-positive and gram-negative. Our peptides are also, this is a transmission electron microscopy image of a surgical dressing on the wound in that mouse model I just showed you. In vehicle-treated samples, massive biofilms form in that wound on essentially every surface. Biofilms are a source of physical resistance to antibiotics and resident microbes that continue an infection long term. So these biofilms are completely absent when these wounds are treated with our peptides. So not only do we kill the infection, we prevent biofilm formation, which is a problem. If we take an organism like Pseudomonas aeruginosa, a very common wound organism that's very good at developing resistance, we find if we passage a normal non-drug resistant strain of Pseudomonas against conventional antibiotics, it rapidly becomes resistant, right? So this is a log scale of the minimum inhibitory concentration versus passage number. And Pseudomonas rapidly becomes completely resistant to all of these drugs. Against our peptides, we see essentially no resistance developing over multiple generations, over many passages. Okay, so I'm not saying resistance doesn't ever occur, but it's very slow to occur relative to conventional antibiotics. So this is the answer to the drug-resistant bacterial problem, is drugs that do not evoke rapid, highly effective resistance. So we have that. So our lead peptide called Q7, and again, I will give you a QR code for publications at the end. Most of this is already published. Q7, it has this host-compatible activity, that is, it's active in the presence of tissue, serum proteins, concentrated host cells, which the vast majority of known antimicrobial peptides are not. It has broad-spectrum activity against the escape pathogens. It treats drug-resistant bacteria in wounds. It treats drug-resistant biofilms in wounds. It treats gram-positive and gram-negative bacteria and gram-positive, gram-negative biofilms, and it avoids the rapid development of resistance. So, you know, I've talked about this. We've shown these peptides are highly efficacious. You know, we have not proceeded to the point of thinking about manufacturing GMP peptide production, but I have worked with a biotech company in the past, so I have contacts in these areas for solving these problems when they come up. You know, we do not see cytotoxicity in cell culture or in the wounds. You know, low potential for developing resistance, and you know, stability. We use non protease sensitive. We use protease resistant D amino acids, so we have peptides that are highly biostable. Even in vivo, they're stable for a very long time at 4 degrees. They're highly soluble, and we've worked on slow release polymer formulations for things like once per day administration. We've shown that they work. So I've created a QR code for some recent Wimley lab publications describing these peptides and the evolution of these peptides that we're very excited about. And that is what I have for you today, and I'm prepared to answer questions.
Thank you. Nice presentation. Have you done any screening to see that there's microbiome sparing effects? Does this peptide target commensals?
It will target commensals if given orally, and that's not the route of delivery that we're considering at this point. Peptides of this length are not bioavailable through oral delivery without some special changes to their formulation. So in terms of the gut microbiome, I don't think these peptides will end up being exposed there. They will kill commensals if they end up in the gut. So we're thinking more of surgical wounds, diabetic foot ulcers, and pressure ulcers, where many of these fatal drug-resistant bacterial infections actually begin. So that's the application we're thinking.
So 2 quick questions. What's the proposed mode of action? Is it membrane disruption of the bacteria? And do you see any immune responses from the peptides?
The mode of action is membrane disruption. And we think this is exactly what is being selected for, is an interaction with the bacterial membrane and lack of interactions with host cells, et cetera. We haven't tested for immune responses here But peptides of this length, these are 10 to 15 residues. They generally don't elicit immune responses. We don't know for sure whether that's the case here. Okay. Wow.